the Evening with Neuroscience coordinator, and I'm your MC for this stream.
Hi everyone, my name is Terry, and I'm the editor-in-chief of Gray Matters Journal.
And I'm Sharda, the senior editor for Gray Matters.
Welcome to An Evening with Neuroscience 2020. We are happy to have you all here with us. This is our first year doing a live stream, so if you are tuned in right now, we're happy to have your support. An Evening with Neuroscience, or EWN, is Gray Matters Journal's event where for the past 8 years we've brought in a panel of neuroscientists from various fields, and this year we're bringing in a philosopher to answer your mind-blowing questions. EWN is truly a community event at heart. It is organized by a group of passionate undergraduates supported by the university and local businesses. The planning for this event started in September, and though we worked tirelessly, we were met with quite the unexpected challenge only 2 months before EWN. We had to make a lot of quick adaptations, and I'm very proud of and thankful for my team for sticking with this project. Our entire team is showcased on their website, and also on our website are our amazing sponsors, which we are very thankful for. Before you hear from Terry, our editor-in-chief, I would like to say a huge thank you to my team, as well as all of the individuals and businesses who have made this event possible.
To expand on what Gray Matters is, for those attending their first EWN, Gray Matters is the University of Washington's undergraduate neuroscience journal. 10 years ago, we were one of the first undergraduate neuroscience journals in the nation, and since then our mission has remained the same, to produce high-quality neuroscience articles that are informative, visually appealing, and accessible to the public. Every quarter, our staff of 60 students collaborates to put out a collection of articles that cover a variety of topics from issues on human health to more abstract concepts like how our brain perceives the world. And because improving scientific literacy and increasing access to science publications is something so near and dear to our team, all our articles are viewable for free online at our website, graymattersjournal.com. Gray Matters has been the cumulative work of more than 1,000 students over a decade. We are so proud of the work that has been put in to make the journal what it is and to have built a platform that allows us to expand into outreach. EWN is currently our largest outreach event of the year, but we are constantly looking for new ways to put on events with heart that help us all learn from each other.
The three of us are graduating this year, and we're proud that EWN is finally happening. I've helped with this event for the past 4 years, and it's been amazing to see it grow. We've gone from 100 people in the Health Sciences Auditorium to 700 in Kane Hall, and although we've had to change our format this year, we're passionate about bringing accessible neuroscience to the public. We've presented about our journal and our event at the Society for Neuroscience Conference to reach more people, and we hope to expand both our journal and our audience. If you would like to donate to help keep this event and Gray Matters running, there will be a link in the description below, and we're accepting donations from Square. Tonight's event will be broken up into 3 parts. We'll start off with panelists answering pre-submitted questions, followed by an intermission where viewers are given time to submit their own questions live. The final segment will feature a discussion of audience questions, and at the end of the stream, we will pick one particularly insightful question or comment, and you will receive a gift from one of our sponsors. All throughout the event, however, moderators will be monitoring the comment section, so if you have any questions, feel free to comment them, and we will choose some of them to ask the question during the last section of the stream. Please remember to keep your comments respectful and professional. There are no bad questions. Feel free to bring forth your wild and wacky ones. As our panel is not practicing physicians, however, we will not be accepting questions about personal medical histories. We hope that you will go away from this event having learned something new. So without further ado, welcome to An Evening with Neuroscience.
Hello, my name is Adrian Casey Lee. I'm Professor of Speech and Hearing Sciences, and I'm tonight's moderator. It's my distinguished honor to introduce our panelists tonight. I'll start with Dr. Susan Ferguson from Psychiatry. Dr. Ferguson, would you like to introduce yourself?
Yeah, sure. Thanks very much, Adrian. So I'm very excited to be here in our new format. Hopefully this works without too much issue, but so I'm in the psychiatry department. I'm an associate professor there, and I have a couple of different hats that I wear as part of my job. One of those is I run a basic science research lab at Seattle Children's, which is located downtown, and we use rodent models to study addiction. And so we focus both on psychostimulants, primarily cocaine, as well as opioids like heroin. And we've also actually tend to study combinations of those, and that's because polysubstance use is actually pretty prevalent among drug users out in the communities. We won't be able to model that. What I'm interested in understanding and studying is how the brain changes to drive the development of addiction and relapse. We also want to know why some people are vulnerable to developing addiction and what makes others resilient. I also serve as the director of the Alcohol and Drug Abuse Institute at UW, and in that capacity, I serve more as an administrator. But our institute is really focused on how to get treatments and out into the best current treatments. How do we get those out to people that need them? Particularly, I guess, looking at sort of underserved communities. These are homeless folks, Native Americans, and incarcerated individuals. And then I think there was a prompt to answer by way of introduction. So my field, as I said, is neuroscience. And one of the questions was, what do we want people to know about our fields? And I think the main thing for me with addiction is to understand that it is a brain disease. So the drugs really do change sort of fundamentally the ways that people with substance use disorders, the ways that their disease, that their brains function. And so it can be very hard for people to stop. I think it's important to understand that it's not necessarily a choice, but it's really a fundamental disease. And I'll turn it back to you, Adrian.
Thank you, Susan. Sarah Goering is our philosopher on the panel.
Hi, thanks very much. So I'm Sarah Goering. I'm a professor in the Department of Philosophy at the University of Washington, where my work is really in medical ethics and feminist philosophy. And in the last 8 years, I've been doing a lot of work in neuroethics, in part because I'm affiliated with the UW Center for Neurotechnology. It's a center that is working on developing ideas for how to do neural devices that would aim to assist people with motor disorders like Parkinson's or essential tremor through deep brain stimulation, but also brain-computer interfaces that are intended to read out from brain activity in order to control something external to the person, such as a cursor on a screen or a wheelchair or a robotic arm or something along those lines. I guess in terms of the things that I wish that people knew about my area, so I think people should really be inspired and excited about philosophical conversation rather than intimidated. And in ethics too, I think sometimes people think somebody coming in to do ethics on a project is really there kind of as a regulator or a police person, ethics police person. And that's not at all what our aim is. Our aim is to really collaborate with researchers and think about the human dimensions of a lot of the neuroscientific research and neurotechnology research that's going on. And maybe quickly, one other thing is I have a hat in the disability studies program at UW as a faculty member there. And I think it's really important for people working in this nexus of fields to realize that there's a vast diversity of viewpoints and perspectives on disability, and many of them don't involve seeing disability as something that's purely internal or in need of fixing, or even if there are things that a person might want to interact with the world, it's more assistive rather than thinking there's something problematic with the individual. And that's just to recognize the diversity of humanity. I'll pass it back to you, Adrian.
That's great, Sarah. And in fact, we'll delve deeper into that question later on. Dr. Zen Kone, it's from neurological surgery.
Hi, everyone. My name is Zen Kone. I'm an assistant professor at the University of Washington, like Adrian said, in the Department of Neurological Surgery. And my area of research is in neuroplasticity. And currently, we're working on spinal cord injury and repair. And we actually, I'm really excited to be part of this panel. It seems like, a philosopher on this type of panel is pretty exciting. I can't wait to hear what she has to say as well. So I'm pretty excited, and I'm looking forward to your questions.
And finally, not least, Dr. Maureen Knights from ophthalmology.
Hi, everyone. I'm Maureen Knight. I'm a professor in the Department of Ophthalmology, and I study vision, not too surprising. And I'm interested in everything about vision, from genes to behavior. So I have co-authored a number of patents that have been licensed to companies. And so I'm also interested in translational medicine, trying to invent something that will cause or prevent some sort of progress, and then getting it out to patients. So So there are a lot of challenges, both in the invention side of things, testing it locally in the lab, and then getting the intellectual property patented so that someone can commercialize it. And so I'm interested in doing all of that. Back to you,
Oh,
Adrian.
thank you. Thank you, Maureen. My name is a bit confusing. I go by KC, or that's my sign name. And before going to the questions, my research is in auditory attention, and in fact, you struggling to listen through a poor auditory channel, but actually also helped by seeing our face at the same time, unless it is lagged. Those are the different questions that we study in our lab and how the brain processes auditory and audiovisual information. So one of the caveats is that before we start, brain is really complex and we may not necessarily have all the complete answers. That might frustrate you, but I think I can collectively say that the fact that there's so many questions out there really inspires us in this field. So without further ado, let me start by asking the first questions we received. So what is the most interesting thing you have ever learned about your research or from communities related to your research? I believe, Sarah, you want to take on that question?
I'll start. It's always hard to say the most interesting thing, but one of them I would say, especially given my field where philosophers don't have labs typically, we don't, need equipment and that sort of thing, is that it's very important to be in touch with people in my area who are using the devices that we're talking about to really get a full and robust look at the way the devices affect the way a person lives. So just as an example, we did a focus group with people who use deep brain stimulation for treatment-resistant depression. And this is the thing, I have to say, going in, I was pretty skeptical about the trial overall. Hadn't worked, so it stopped partway through. But of course, within those numbers, there were some people who had done really well with the device. And when I went into the focus group, I was surprised to hear from people who, granted, were well enough to come to the table, that it had saved their lives. They really all around thought that it was an incredibly valuable intervention. So I was surprised at that. But then also just to realize that sometimes when we use a neural intervention to help somebody, it can treat and really help one thing, but then create different ripple effects for that person in different areas. And I just think it's really interesting to pay attention to that. So again, as an example, some people felt like their depression was much improved with the device, but then they had interesting relational effects with family members where they didn't feel like they were being treated in the same way as they wanted to be treated, or people even used language that they felt dehumanized. And this is just not something I would have expected. If it's dealing with the depression, you might think things will just move along then, but it's just so important to really listen to people who are, I think, in many ways marginalized otherwise, whether it's because of the disease or condition or just their circumstances, to make sure that we get those voices and include them in our philosophical and ethical thinking. So that's how I would answer that. Back to you.
Awesome. Well, the next question is really interesting. Do you have a favorite type of neuron? What is it and why?
Well, that's like, everyone should chime in on that one. So I'll take the first dib on that one. So my favorite type of neuron is the pyramidal neuron. Yay. Right? So, it's a really cool type of neuron with lots of arborization. And one of the reasons I really like it is because it has lots of, one pyramidal neurons can have 20 to 30,000 inputs, right? Synapsing onto its dendrite. So it's like a little processing unit, you can think of it as. And it's also an output neuron. So that means that it sends whatever information it gets to that neuron, and then it processes it, and then sends it out to the other areas of the brain. So it's also really pretty on histology and staining under the microscope. And I also have to add a little bit, in addition to having a favorite neuron, I'm also really crazy about these cells. They're not neurons at all, but astrocytes. because I study injury and plasticity and how the adult nervous system respond to injury and in aging. Astrocytes are super duper important. They play multitude of roles, from normal blood-brain barrier activities to ion balance in the extracellular space so that neurons can have proper transmission, to having lots of extracellular matrix molecules. And in addition, of course, they respond to injury and assault to the central nervous system. So those are my two favorite types of cells. Back to you, Casey.
Yeah, awesome. I bet the next two questions are for Maureen. So the first one, why are people colorblind? Is it dysfunctions in the eye or how the brain reads signals from the eye or both? Is colorblindness similar to actual blindness?
Okay, so I'm going to answer part one of this. Is it a dysfunction of the eye or the brain? And so most commonly, colorblindness is a dysfunction of the eye, although people can have a head trauma that causes them to lose complete color perception, and that would be cortical blindness, but that's extremely rare. The common forms of color blindness are red-green color blindness. They're extremely common. 1 in 12 males is affected, and 1 in 230 females is affected. So even though everybody says, oh, women aren't colorblind, the fact is it is the most common single locus disorder in females. People are colorblind because they're missing one of the color receptors in the eye. So normally people have 3 different types of receptors. And if we orient ourselves to the rainbow, where we have red, orange, yellow, green, blue, and violet, red is at the long wavelength end, blue is at the short wavelength end, and the other colors are in the middle, with green being kind of really in the middle. So in the human eye, there's one receptor that is sensitive to long wavelengths, another one that's sensitive to short wavelengths, and another one that's sensitive to the middle wavelengths. And so people are colorblind because they're missing a gene that makes a protein that is responsible for absorbing light in either in the long or the middle wavelength region of the spectrum. And this gene is on the X chromosome. And so females have 2 X chromosomes, males have an X and a Y. So males are always colorblind because if they have the genes, if they're missing a gene for one of the receptor types on the X chromosome, they will have color blindness. Females will only have color blindness if they have the same type, they've carried the genes for the same type of color blindness on the same X chromosome. So that is why people are colorblind. And then there's a common misconception. People say, well, so if people are missing one receptor type, either the middle or the long wavelength one, they can't see any of or distinguish between any of the colors in the red-green region of the spectrum. That includes yellow, green, and all the intermediate colors. And it's because the way the information is processed in the retina and in the brain. Sometimes people who are colorblind will name colors correctly, and it confuses people with normal color vision, and the colorblind people as well. They go, well, I can name colors just fine, but they're not naming colors based on the color alone. People often can get the names right because there are brightness cues, like yellows tend to be brighter than reds. And so people that are colorblind learn all these other cues that aren't the color to help them name colors correctly, even though they're not seeing the same color differences that we're seeing. So then the third part of this question is colorblindness like blindness, like actual blindness? And the answer is yes, it is. And that's why we call it colorblindness. That in the most severe form, which affects about 2% of males, there are things in the world that are invisible to them, that are clearly visible to people with normal color vision. And in fact, this is how we design color vision tests. If you've ever seen some of these tests, there will be a page with a bright pink square and a similar bright pink circle, and a colorblind person that has will look at that and say, oh, I see a circle. And the parents of these kids will be looking at it too, and they're going, but what about the square? And the kids are like, there's no square there. There's just no square there. But it's just overwhelmingly this big bright square. So there are things in the world that are invisible to them that are visible to normal people, and in that way, it's like blindness.
Wow, Maureen, you've given such a great answer. You've almost answered the next question, so I'm going to skip the next one about the similarity between blindness and colorblindness. I'm going to briefly take on the next question. Nowadays, there are hearing aid companies that actually try to read brain waves such that they can selectively amplify sounds of interest. So hearing aids right now, it's really hard for people with hearing impairment because they amplify all the sounds. But we seamlessly, selectively attend to, for example, the male speaker, and we can de-emphasize the female speaker. But if you're just using a hearing aid, that's really hard, for example, in a crowded restaurant. And the question really is, how would attention disorders like ADHD impact such hearing aids? That's a great question. In fact, that's a whole question on neurodiversity. When we study neuroengineering products or neurobiological solutions, we have to take into account of all the different uses at the other end. And so that's a great question. We don't know. And in fact, we need to know more about brain signals that's coming from diverse populations such that we can actually design it better. Even for the hearing impaired subjects, we don't necessarily know that the different kinds of hearing impairment along the auditory pathway, how that changes the brain signals yet. So having that device out there would be great to do the field testing, but there are many fine-tunings that needs to be done in the coming years. So thanks for that question. All right, so moving on. Next question is it true that with the lack of one of the five senses, the others would become stronger, or is that only because we have to rely on them or use them more?
So I'll take a stab at it. I'm sure the rest of the panel can also add to this. But, the short answer is yes. The other senses do get stronger, but stronger is an interesting word, right? So is it the synaptic strength, the connection that we used to have? Does it get stronger? So in that sense, synaptic stabilization, synaptic maturation, and synaptic strength, I would say, can increase. You can also have areas of the brain that used to process say a sensory input like touch, use it, and then other areas of the brain take over that area. So in that way, you can have plasticity not only at the cellular level but also at the anatomical and mapping level. So the short answer is yes. It's true that you can have stronger, but we also know that other brain areas get taken over when we lose a sense.
Thank you.
Add to that?
All right, so sorry. Awesome. So we're sort of moving gears because this is an interesting segue into timing a bit of philosophy and ethics. So as we're talking about deafness and blindness, and in fact these terminologies are starting to evolve, is it ethical to conduct research to cure certain diagnoses like deafness or blindness when maybe the people in those communities aren't looking for a cure? For example, community with deaf with the capital D. Susan, do you want to take a stab first, and then Sarah?
Yeah, definitely. So I was interested in this question too, because I think it really applies to many diseases, not just deafness and blindness. And so one of the things that I would say is that the goal of most research into diseases is to really try to fundamentally understand what causes them, and as well as how we can go about fixing them, how we can develop a cure. So I think that research should go on, I guess, irrespective of whether someone's interested in being cured by a disease or not. That's just sort of our basic understanding, to be able to then offer up cures as an option. So and Sarah can certainly comment more about the ethics around it. So I think it would be unethical to impose a cure on somebody that doesn't want one. But I think that's different than what we're trying to do, which is really just develop cures for diseases. So, there's really a lot of diseases out there that people don't necessarily want cures for, that we, have been developing them. So one example that came to my mind is cancer, especially for sort of late stage cancer people. A lot of people just, choose to not go with the treatment, right? And the same is true, in the area that I study, which is addiction. Many people don't want to stop using drugs. And so I don't think worse cure or treatment at all, but I like, from a science perspective, being able to offer them as options to people. So I don't know, Sarah, if you have anything else to add.
No, I think that's a good start. I think one of the things that I would add is just that it pays to know something about the range of people who would otherwise qualify as potential people using whatever intervention that you're developing. And I think knowing that just changes the way that we talk about some of the things. So if you think about something like cochlear implants, knowing that the capital D deaf community is there, and there are people who are perfectly fine with their bodies, with their interaction with the world, or wishing that there were better accommodation but not needing a bodily fix, just knowing that might alter the way we talk about the technology under development, that it's an assistive device, right? It's a thing that can grant you a different kind of access to the world, but it's not that you're necessarily deficient and in need of being fixed. It's that here's a different alternate way to get access to the world. And I think, in the world that I work in, thinking about neural devices too, having talked with some of the PIs, I think there is just a way in which when you start thinking about who the end users of your technology may be, and the diversity of perspectives on what they would most like to have you working on, if you really care to help them, then you just open up the range and of what you'll work on, for one thing, and you'll probably talk about those individuals with a greater degree of respect, just recognizing those differences.
That seems like a great segue to, Maureen, would you like to add something?
Yes, I would. Because in the field of blindness, I mean, I've actually been faced with this issue in a weird way. So I, like Susan was talking about, had worked on a cure for colorblindness as a neuroplasticity question. So I worked on it in a primate model, and it was very, the experiments was chosen by Time magazine as being the #3 most important scientific discovery of 2009. This published result that we could cure colorblindness in a squirrel monkey, or in a primate model that had it was an exact model for human colorblindness. People want some colorblind people say, oh, well, it's not a problem. Why would you have such a cure? And there are other people that say, can I come to your lab? I know it's not FDA approved, but you could just give me the injection and I could be cured, because there are people that have accidentally killed people because they didn't see a signal. There are people that have not been able to have their dream job. And so there, in the field of vision, there's clearly people that don't want the cure, but there are clearly people that do want the cure. And even to the extent that these people form their own foundations to fund research, to develop cures that can help their people that have their rare orphan disease. So it's a really interesting question. I'm not sure that there's an actual right or wrong answer.
That's fascinating. And in fact, a follow-up question to this is, what is the biggest way neurodiversity will impact your research in the next 5 years? There's, in my mind, they're interrelated. Zen?
Sure. So I study plasticity and how the spinal cord injury repairs itself, or how ways in which we can augment that repair process. So in that, we certainly see lots of neurodiversity in a sense, where a lot of different animals, for example, may be able to reach for a particular task, like reaching for a sugar pellet, for example. The animals really love to do that. So that's one of the behavior that we study. And it's sort of equivalent to human beings being able to reach for a cup of tea, or a glass of water. So really important aspects of injury and plasticity. And if you think about it, does it really matter how we are able to figure out how to reach? We're going to use all the leftover circuits, whatever is spared after injury, to do that. So in my research, we really think a lot about different ways in which a task or a function can be achieved using multiple different ways of the network can reconnect itself. So this sort of idea of, different ways of learning and different ways of achieving a particular function is very important to my research. And not just the last 5 years or the next 5 years, but it's, really an ongoing theme. And I think it will also play a significant role if we think about all the different ways in which a neural circuit can rewire. We can also think about how, and have we been really rigid about, doing rehabilitation, for example, just trying to do the same motor task. Is that the right kind of rehabilitation to do? Or there are some other general motor ones that we should be doing? So back to you.
Sarah, I think this is the question for you. What do you think the biggest policy change relating to neuroethics will be in the next couple of years?
It's a hard one to answer. I think one of the things we're really going to have to think about is how we think about neural data and privacy. Because I do think that we've given up so many parts of our private lives through our interactions online and social media and through, purchasing and that kind of thing. And I think the more neural data we are able to collect, and then the better we get at understanding what we're looking at, and then we'll be able to pair that with a lot of this other data trail that we already leave around us, we're going to have really interesting and more pressing questions coming up about how we protect our private space. I think there are some ways in which the things that go on internally in our heads are the most private thing that we have, that we choose how we share them now. And I think there's a real worry out there that a lot of those things are not going to be as private. So we have to figure out how we put together policy in order to protect those things. And even though many people, I think, will say, oh, well, it's privacy, I don't have anything to hide. I don't think it's about what you have to hide. I think it's about the importance of having this space that you have control over. In our language, we'll talk about negotiated access, where you can figure out who you let in and who you keep out. And it preserves a kind of intimacy so that you're allowed to maintain the kind of intimacy you want and share when you want. And I think right now what's happening is we give up a lot of that, maybe without fully realizing what we've given up.
Thank you. I think this is a question for Susan. So are you ready? Can you talk about the neurology of addiction recovery? How do our brains change when we're trying to get over drugs? And what is the difference between cold turkey, slow quitting, from a neurological standpoint?
Sure, yeah. I was also just thinking about what Sarah was just saying, and just thinking about how with COVID now and a lot of the tracing that they're starting to think about having to do, and how that from an ethics may or may not present sort of an interesting ethics dilemma. So I think there's a lot to be said for philosophy around many ethical decisions around technology these days. But yeah, with respect to the question that you just asked, Casey, so I think there's really multiple stages to addiction from a brain perspective. And so, what we really need to always keep in mind is when you take a drug, it's going to have both sort of an acute, immediate effect. And this is true whether it's a drug that people can misuse, or even just an aspirin, a Tylenol, when you have a headache. And then when you take drugs repeatedly over time, there can be a lot of long-term changes that are taking place. And we've touched a little bit on the panel today about something called neuroplasticity, where the neurons and cells in the brain can actually get rewired. And so those long-term changes with addiction, they often take place in areas that regulate motivation and decision-making. And so when we're thinking about getting off of drugs, we also need to think about the acute phase and then the much longer long-term recovery phase. So the acute phase, which I think kind of comes into this question about cold turkey versus slow quitting, is very important from a medical perspective. So with some drugs, when people are very dependent on them, so some examples are opioids, and with alcohol, if you just completely stop drugs, you can have very serious neurobiological effects, and you can die from that withdrawal of that drug. So this is, like I said, particularly true for alcohol, where you wouldn't just want to stop. You would want to very slowly taper off. The same is often true, like I said, with things like opioids. With other kinds of drugs like nicotine, you're not going to die from getting off of the drug. But with all of those drugs, when you think about them, your body being used to them, maybe the best example is something like caffeine, where people know if you just kind of stop all of a sudden, you can have sort of physical effects that happen. Things like you get a bad headache, you maybe have some sort of hangover, that sort of thing. And so, like I said, the severity of those symptoms can vary a lot, but that's really just kind of getting the acute drug out of the system. So that's the initial phase that's very important. Someone on sort of the detox phase that we talk about, where you're just kind of getting everything out of the system. And then what's probably in some ways I would consider to be, I don't want to say more important, but for long-term recovery, we really need to think about what are the long-term changes that have been taking place in the brain, and how do we sort of recover from those, if you will. And so, like I said, this is a complicated question. I don't want to take up all the time on the panel, but, I think, so just to say a few things, it really depends on the types of drugs that we're talking about. So some drugs, there's going to be permanent changes that take place. Methamphetamine is an example of this, where you can get sort of permanent structural changes and brain changes that aren't necessarily going to recover. But you can use things like behavioral therapies, different kinds of things that maybe rewire some of those connections. And then you also have other kinds of long-term changes that over time will start to go away. So we've seen this sometimes with cannabis, with marijuana, where over time, some of the initial changes, some of the decreases in, let's say, dopamine release or dopamine neurons, same thing with psychostimulants, over time, those kind of rebound and can get better. So I guess, like I said, it's a complicated question. Hopefully that gives the audience some sense of things.
Awesome, thank you. My next one, I actually wonder whether my...
Before you start, we would like to pause briefly for an interpreter switch. And we will let you know when that is done. And the switch is done. Holly, would you mind turning off your video? Thank you.
All right. I wonder whether my wife submitted this question. So we often talk about how to improve memory, but what about the other way around? How do we work around forgetting things, such as trauma? Zen?
Well, this is a really interesting question, really exciting questions that I think about actually for everyday life as well as research, right? So from the philosophical standpoint, thinking about memory, would it be good to remember everything? I have 2 children, so I'm not sure if I remember everything about my first childbirth, would I actually choose to have a second one? I'm not sure. Also, if you're really interested, there's a book by Borges, the story of Funes the Memorious, where it's a story of a man who fell off his horse and had brain injury and came up with this condition where he remembered everything. Think about that, right? Me now, me 10 seconds ago, and me the next 10 seconds. I mean, it could drive a person crazy, right? So remembering everything might not actually be good. And certainly there's room for forgetting. Again, sort of the question asked, what about bad memories and trauma? And neuroscientists have been studying that as well. And you can think of forgetting or suppressing, a bad memory or traumatic memory from association cues, like the sound of a bomb or a bang that sort of, produces stress responses in soldiers, for example. So it's the idea, I think, is called extension learning, where you're actually learning that cue doesn't actually need to have that stress response anymore if you're not in the war zone anymore. So that sort of idea of extinguishing memory, and it's sort of like not forgetting, but the idea is you're overlaying on top of what you've learned before. So learning new things.
Awesome. The next question is, what are the pros and cons of using behavioral therapy as a treatment for addiction versus drug therapy? Susan, would you like to take on that question?
Yeah. So thanks for that. So I think, again, as I was sort of talking about in my other question, this really depends a lot on the kinds of drugs that we're talking about. So there are some drugs, and some classes of drugs. So psychostimulants would be an example of this, cocaine, amphetamine, where we actually don't have any drug therapies. All of our therapies are really behavioral. And so in that sense, I guess the con would be, trying to have some sort of medication assisted treatment that could work and sort of help integrate with those behavioral therapies would be really great. And then there's some drugs like opioids, where we actually have some pretty good medications. They're not perfect by any means, but they can make a world of difference for people. Things like methadone and buprenorphine. So something like methadone, just to give you a sense of what it is, it's an opioid and so what that means is it'll bind to the same types of receptors in the brain as something like heroin and morphine, but it's kind of like if you think about smoking a cigarette versus a nicotine patch. So smoking a cigarette, you get brief bursts of nicotine into the brain, whereas if you put a nicotine patch, which is also a medicated assisted treatment, you get very low levels of that same drug into your bloodstream that lasts for 24 hours. And so when you have a very low level of a drug in the brain, you often don't get those the rush, the spikes of drug reward, the cravings, all of that kind of thing. So with something like methadone, you get the highs and sort of the out of control behavior that you might get within a different kind of opioid, with the cravings that come along with an opioid. And so those drugs can be very powerful for sort of dampening down craving. But in and of themselves, even for drugs like methadone, they're not a cure and they're not a perfect treatment. And so I think in the fields, we've really found that combining any sort of drug treatment with behavioral therapy is really kind of the best option in terms of helping people manage the choices that they're going to make, help sort of rewire their brain and thinking about the consequences of things. So, and I just want to acknowledge that I use that word choice, and I had said before that we can't really think about addiction as, a pure choice. And what I mean by that is that it's our brains are rewiring. So we can have behavioral therapies where people might get an urge to use a drug, and then the behavioral therapy can kind of come in and help them to sort of think through why that urge, ways perhaps that they can explore that to dampen down that urge. Just like I'm having the urge right now, my dog is growling in the background. I'm having the urge that I'm trying to sort of dampen down to go calm her down. But I think, so from my perspective, that there's really, there's not necessarily a pro or con of either. It's, I think the best approach is a combination approach of using all of those therapies.
Well, thank you. That was a great comprehensive answer. We're going to switch gear a tiny bit. There are a few questions that are related to career or interdisciplinary research. The first one is, what is your best advice for college students considering research careers? What are ways to be involved in research other than academia? If you could study one other field, what would it be? I think Zen wants to take this question up.
Sure. So what's the best advice for a college student? I would say stay absolutely curious. You never know what kind of field you're studying is going to provide answers to the research questions. So I started out as a neuroscientist, and I'm still a neuroscientist, but I have incorporated in my research, at least, lots of areas like neural engineering and just bioengineering approaches to my research. And that's been really exciting. So, lots of interdisciplinary things are happening that helps us to answer very specific neuroscience questions. Math and physics has always been a part of, understanding how the brain works. And so I think stay absolutely curious. Like I said, you never know which classes and which areas of your studies are going to bring you insight to what you're wanting to research on. Now, the other part was ways to be involved with research. I would say, academia is great. Obviously, we're in this institution, amazing, and have a lot of opportunities. So that's definitely is an area that I would encourage you. But other than that, like the Allen Brain Institute, it's not an academic purely institute, but they also do really interesting research. National labs are really interesting. All of those different institutions have different goals, very directed goals. So those are areas that you can look into. Industry, obviously, is another choice that you can look into. And all of those have very different specific goals that they're trying to achieve with their research. But, those are all different flavors of research that you can explore. And if I could study other than neuroscience, I would have to say immunology. It has always been fascinating to me. But I probably would be a neuroimmunologist, which is what I am. So I'm kind of straddling a couple of those areas of research, I would say. Thanks, Casey.
Awesome. Now, Sarah, I've got a question for you. The question from from what we got the web is there someone you would like to collaborate with from the university or in your field? I'm interested from hearing it from a philosopher.
Sure. So one of the greatest things about my work right now is that I do get to collaborate with neural engineers and neuroscientists through this Center for Neurotechnology. But one of the areas that I'm really interested in getting into is working with people who are looking at neural devices that will help to give sensory feedback, in particular tactile sensory feedback. So for instance, if you can be a user and basically use a brain-computer interface to, let's say, drive a robotic arm, if we could put sensors on that arm so that you can feel pressure and shape and other things just through the hand without your visual input. I think it's really interesting to think about the effect of that on how we feel where we are in the world, especially if we don't have our visual input going, and just the ways in which we learn to trust our place in the world. And then beyond the neuroscientists or neural engineers, I'm also really interested in some of the work going on at the UW Law School with Ryan Kalo in the Tech Policy Lab, which we bump up against each other in various ways, but haven't really had the chance to interact more fully. And I think there are a lot of really interesting philosophical and ethical issues that then end up looking slightly different when we get in the realm of policy. And I'm just curious about how, what needs to change in making policy.
Awesome. I might take on the next question. So the question is about the interaction between neuroscience and other fields of research. I myself was trained as an engineer before somehow fell into neuroscience. And so the field of neuroengineering, the combining, making things happen by understanding neuroscience and leveraging the knowledge of both, it's an exciting field. And UW is especially good at it with a lot of experts in this field. So for those that are around Seattle area, please explore all the neuroengineers in the area and get your hands dirty. Understanding neuroscience is exciting, but being able to make something out of it and for the betterment of mankind, I think that's what we engineers believe that we do. It's an exciting marriage of both fields. Zen, do you have anything to add?
I would totally agree with that. I think sometimes neuroscientists, as a biologist or neuroscientist, I really want to understand all the mechanisms and all the neural networks that underlie a lot of behavior or function that we're studying. But at the end of the day, we're here. Our research hope to really not only gain new information and learn new things about neuroscience and have better understandings of how our brain works, but also to really help patients and people and have our better interaction with our environment. And so I think, collaborating with neuroengineering folks have been amazing. I actually did my postdoctoral work in an engineering lab. So, really immersing myself in engineering because engineers really know how to get things done. Right? They just want to get from point A to point B, and that's really awesome. Sometimes I think neuroscientists kind of get lost in just trying to discover what is the actual underlying things, which they are very important for, understanding the network better. But patients with spinal cord injury would like to figure out how to make their bladder work, or so there are very important questions that we need to answer, that we don't necessarily need to know exactly how the network is rewiring, as long as the person can, with neuro devices or interfaces, able to reach that cup of coffee or put a fork in their mouth and feed themselves. That's a huge life-changing advancement. So I think there are lots of areas of overlap in engineering and neuroscience, for sure.
Well, as an engineer, I'm also particularly good at keeping time. So I'm just looking at time, and we have one more question that we want to sneak in. Maureen, are there drawbacks associated with using induced stem cells rather than embryonic stem cells? I think that's an interesting question to delve in.
Yes, there are some drawbacks. And the main drawback is that the protocols used to induce adult cells to become pluripotent stem cells are mutagenic. And so what people have found, there are a number of clinical trials ongoing where people are transplanting retinal pigmented epithelial cells into patients that have various kinds of vision disorders, degenerative vision loss. And they, what people have discovered is that whatever the protocol is that causes a cell to be induced to become a retinal cell tends to cause mutations in tumor suppressor genes. So now people are characterizing these cells that they're implanting in patients and realizing that they've got to tell the patients, now we're about to do this surgery to implant these cells, but we have to tell you that it has a mutation that could cause a tumor to form in your eye. And do you want to go ahead, or do you not want to go ahead? So that's one of the main drawbacks, is that the protocols to induce them to become adult cells to become stem cells is mutagenic.
All right. Well, thank you for this concludes our first half of the panelist discussion. There'll be a 10-minute break coming up. And during this time, please write down any questions that you might have for the panelists. And I believe that you can post it online. You'll notice scene change. I might actually have to change rooms so that my kids can come to my room and do the night routine. So see you back at 8:05 p.m.
one, go.
All right, welcome back. Hope you had a good break. Change of scenery. I'm now in my kids' room. And so we're going to start with the first question submitted by Miranda. What are some ways we can help our brains during quarantine that we might not have heard yet? We're all discussing among the panelists. Don't we all want the answers to this? Does anyone want to take on this question?
Yeah, so this is Susan. I'm happy to give it the old college try, I guess. So I certainly don't have all the answers, and I'm not sure that, these are necessarily new, but I think, one of the most important things to realize and to keep in mind is that we are in a pandemic. It's not a normal situation. Even though we're all at home, it's not that we're just at home because we were sick and didn't want to go into work or go to school, right? So things are not normal. And I think it's recognize that and sort of recalibrate our expectations around a lot of things. I think that's really good for our mental health. And so it's okay to not be as productive as what you would normally be. And I think we all need to kind of embrace that. And so I think that's very important. I also think it's really important. I hope people are setting boundaries around things. And so what I mean by that is because so many of us are at home, we need to still sort of create some of those norms. So for me, it's around work, right? So I might be inclined because Monday isn't really any different anymore than a Saturday. So why not just work? So I think it's very important to still maybe focus on work during the work week. And then during the weekend, maybe put down that computer, set aside the email, focus on being outside around your homes, doing different kinds of activities that you can still engage in that are separate from work. I guess I would also add, to really set realistic expectations. This is around the idea that we don't necessarily need to be productive as productive as we would be otherwise. And so, even if in a day you only get a small amount of the things done that you wanted to do, then sort of be content and that's okay. I think the other thing is, about trying to stay connected with people, even though physically we have to be, separate. The idea that we can still, this idea of socially distancing ourselves is more of a physical thing. So kind of like what we're still doing today, I think it's important to still do the things we would have otherwise done in person and figure out a way to kind of still do them apart, whether it's a lot of Zoom meetings now. I know there's a lot of people kind of doing these Zoom happy hours and Zoom family time and that sort of thing. So I guess that's kind of the ideas that I had. I don't know if anyone else in the panel had anything to add.
Can I just really quickly, I don't have an answer here, but what you said reminds me of what a physician friend of mine keeps saying every time I call him, which is, it's not social distancing, it's physical distancing. Why do we keep using this language? We should be remaining socially connected insofar as we can. So I just thought I'd share that. He'll be very proud of me.
Awesome. We might move on to the next question submitted by Jacqueline. How do other senses improve during sensory deprivation or after having lost sight, hearing, etc.? Or is that just a myth? Zen, do you want to take on that question?
Sure. So I was going to tack on to the last question with the panelists about, staying healthy and during the quarantine. I would just also make sure that everyone focuses on self-care. We're kind of trying to be so productive, but at the same time, can't really be productive if you're not taking care of yourself. And that's your sort of mental health note to take care of yourself. And this kind of activity we're doing, we're all coming together as a community, as neuroscientists or just as scientists, and talking and having stimulating conversations really help. So regarding how do the senses improve during sensory deprivation? So I can think of one very specific experiment that I'm very familiar with where, in your cortical representation maps, in a mouse model or a rat model, the little whiskers that the rats have, there's actually a map of each whiskers, and then the sensory input from those whiskers having a represented set of neurons that process that information coming from those little whiskers, right? And so in an experiment, you can actually remove or cut a few of the hairs on that whisker, and you can actually see the other inputs taking over the area that used to represent the hair that was lost. So that is the sort of basis and the idea and evidence for how different neural areas that used to process information of a type of sensory input can be taken over. Now that's within the same, sensory topographical mapping from whisker, one little whisker taking over the other area that the other whisker used to be. But that also occurs with different senses. So areas, if you became, blind, for example, right? So areas that used to do visual processing might then take over other sensory processing. So in that way, we are starting and getting to understand how the brain rewire itself so that you have heightened sense, so to speak. So definitely not a myth.
Awesome. So Jay has a question for Maureen. Given your experience researching nearsightedness, what would you say is the best way we can reduce it, especially if it's genetically caused? How successful is LASIK as a therapy?
Okay. Before I answer that question, I want to just make a comment on the previous question, and the reason is because I just chaired an NIH workshop on curing blindness using stem cells, and one of the things that came up is that so we tapped into the people in the auditory area because we really needed some help. Those people that do cochlear implants, they have a lot of information that we think that could be useful to us. And one of the things that that one of our auditory colleagues told us is that this is a real problem with cochlear implants, that the auditory areas do get taken over by the visual areas of the brain, and that people that get those cochlear implants, that they have a poorer outcome the more they've had a takeover of the auditory areas with the visual areas. And of course, in vision, it's known that visual areas, and you can see this on functional MRI imaging, you can actually see that areas of the brain that should be responding to vision are now responding to auditory signals. So this is an issue. And so now to the myopia or nearsightedness question. One of the things, the best way to treat it is by these glasses that we've just developed. So nearsightedness is a problem that is caused by overgrowth of the eye. So in the visual system, when kids are born, their eye is about 35% the size it's going to be when they're an adult, and over the time that you're a child through puberty, your eye grows in the axial direction, and it's supposed to stop when it gets to a length where the eye length matches the power of the optical components. If it continues to grow too long, that is what causes nearsightedness or myopia. And so a lot of people say, well, who cares about myopia? You just give kids glasses, or they can have LASIK and have it surgically treated. But now, problem solved. But the problem isn't solved, because what the world is having an epidemic of this myopia where it's pathological. The eye grows way too long, and people go blind from secondary problems from the stress on the retina. They have retinal detachments. They're at much greater risk of glaucoma. So this is a really serious problem in terms of the social burden of the cost of people who are going to go blind pretty young in life. And so LASIK is a method of treating the refractive error, but it doesn't treat the underlying problem of the overly long eyeball. And so the way to treat it is to stop the eye from growing in children. So we have invented glasses that have just completed 1 year of a clinical trial that worked, and there are other companies and other research groups that are working on ways of interfering with the signals that are making the eye grow too long.
Awesome. So the next question is submitted by Daniel. In terms of research and designing for people with disabilities, what are the aspects to remember from both an ethical and practical standpoint? Sarah, would you have a go at that?
Sure, I'll have a quick go at it. I just think it's important to reach out in whatever way is possible within a research group. So some of that can be through literature review and trying to understand some of the studies that are already out there. I think about, in my world, some of the brain-computer interfaces that are designed to help with mobility for people with spinal cord injury. You need to know that, what are the priorities of many people with spinal cord injuries? And I think for many non-disabled people, it's a surprise that it really is more about bladder and bowel control and sexual function than it is about walking, right? Even though you might think, but that's the part of the body that seems visibly not to be working in the typical way. And I think there are different ways to do that, right? So you can look at studies that have already been done, qualitative or quantitative ones, and are out published in the literature, or maybe even better yet, so that it's specified to the kind of thing that your lab is working on, is to try to, bring in some people. When we were thinking about some of these brain-computer interfaces, we put together a focus group with people with spinal cord injury in Seattle, not who are already interested in the device, but just who've been living with spinal cord injury. What do you think about these devices? Let's tell you a little bit about what's coming and, get your impressions of them. What are some concerns you might have? What are trade-offs that you think are relevant for how we design these devices? And it seems to me that that's a way that you ensure that you're addressing real needs and that you're designing something that isn't really cool but might be abandoned or not adopted at all, right? It seems to make good sense, but I think it doesn't always happen until the very latest stages of a device. And at that point, there are a lot of sunk costs in that device.
Incorporating design thinking is definitely an important aspect, and I think that's a new wave of thinking for a lot of engineers and entrepreneurs devising new things. Zen, do you have anything to add as well?
I think I definitely second that sort of notion. When I first started working on spinal cord injury, I was thinking about motor and being able to help people walk again. And then talking to patients with spinal cord injury, certainly on the top list is, bowel, bladder function and sexual function, which was surprising. Well, not surprising to us, right? The bladder complications used to be the #1 killer of patients with spinal cord injury. So it's not at all surprising. It's just that we're not seeing it every day when they have to cast themselves. So really real world problems, and having scientists not being just in the lab and really focusing on neuroscience questions only, but talking to people who we hope to have eventual, sort of influence and help on, is really important. So really communicating with the patient population is super important. And thinking about, people trying to help people with disability, or even talking to patients with spinal cord injury. The other philosophical sort of question that I think about is, should that affect my research, or should I be doing research that I'm passionate about, that I'm interested in? So, the push and the pull of that is really important, to be in the middle of it, so that we can kind of really hone in on the most important thing, as well as the most interesting and ways in which we can affect outcome.
And thank you so much. And we'll just do a quick interpreter switch. And we are good to go.
Oops. All right, I'm back. The next question is submitted by Tyler. Is there something you wish the public knew more about from your specialty? I believe Susan has already jumped on the camera and wanted to answer this question.
Yeah, I thought it'd be fun to answer this question, not so much about talking about the diseases that I study, but sort of talking about the work that I think many of us do, and certainly I do as a neuroscientist in the lab. So I don't think people always appreciate, perhaps, or I wish they could all come into the lab and check out all the cool techniques that we have for studying the brain in our animal models. So I would think that a lot of people might be surprised to find out that we have things that are called optogenetics and chemogenetics. So we have ways to actually put artificial receptors and channels in the brain where we can target a specific cell population and actually change the activity of a very tiny part of the cells in the brain or a large portion of the cells in the brain, we as the investigators, the researcher, can turn down activity and turn up activity, and look, and that's one of the ways that we have to look at what these cells are doing in our different kinds of diseases, decision making, all kinds of different behaviors. In addition, we now have tools. One of the things is called fiber photometry, but where we can actually start to image in real time the activity in the brain as an animal is performing a behavior. And so we can watch, it's basically a fluorescent signal. So we put in a calcium indicator. So in our brains, we use calcium when we are performing different kinds of functions. We'll have increases in calcium in the cell, and we can actually measure that and in real time watch that calcium activity change. We can also do it with neurotransmitters now, dopamine, glutamate, serotonin, and we can actually have an animal in a behavioral situation performing a task, and let's say they lever press to get a sucrose reward. We can see in any cell in the brain, we can actually watch and see if a signal goes up or down during that behavior. And I think from my perspective, it's just pretty amazing. And I think it's revolutionizing our ability to understand in animals how behavior, what is happening in the brain to drive behavior. And so I think that's a pretty cool thing that I wish all of the public could come into our labs and check out. I don't know if anyone else has anything to add on.
Zen, did you have anything to add?
Oh, I was just gonna add on that, neuroscience has some really very cool new techniques, but also just generally the idea of the public thinking scientists as sort of the mad scientist personality. I think that's also another one of my pet peeves, that we're just, normal people with all different kinds of people doing neuroscience research, right? And I think it's really bringing people into the lab really makes them see, what we do every day. And in addition, the other one is spinal cord. I do a lot of spinal cord work, and it is part of the central nervous system. So a lot of folks start to really think of it as, the spinal cord as not part of the central nervous system. So although there are lots of differences between brain and spinal cord, it is part of the central nervous system. And that would be really cool thing, those two things. Thanks.
All right. So Valerie had a question in response to Tyler. Building off Tyler's question, are there any common misconceptions about neuroscience you wish the public knew better about? Does anyone want to take on this question? General misconceptions in our field? We definitely use more than 10% of our brain. That would be a misconception there.
And I would just I would just add on to what Zen was saying. I think that misconception of us being a certain type of person, that's just that sort of crazy neuroscientist or crazy science kind of ideas, that we are all people with lots of different kinds of interests, lots of different backgrounds. A lot of us are on Twitter, different kinds of social media, and we have lives outside of science.
All right. If no one wants to jump on this question, the next one, I find it interesting. How do phobias form? This is submitted by Terry. What explains extreme irrational fear? I'm interested to know the answer. So anyone from the panel wants to have a go at this question? I'm supposed to give that awkward silence 5 seconds to brew just to make sure there's not a lag. All right. Well, Terry, it sounds like that you've hit on some questions that stumped the panel. Unfortunately, we wish we know all the answers, but this one, oh, nope. We have a takeaway.
Actually, Adrian, so I'm going to jump in. I only know a tiny bit about this, so I'll So I don't want to speak too much about it, but I do know that there is some research to suggest that they're passed on from generation to generation, that you don't necessarily have to have encountered the scary thing to have a fear of it, that there are some sort of innate responses, fear of spiders, snakes, those sorts of things. So I think that's about all I'll say about it, because I don't know in depth about it, but it's pretty interesting that you don't have to have encountered the thing to have a phobia of it. You can thank your parents.
Or now I can blame myself for how my kids behave. The next question from Miranda. How has studying neuroscience changed your outlook on the world, or how you interact with the world outside a purely scientific context? I'll have a go at this first. The more I study neuroscience or science in general, the more I appreciate what I don't know and what we don't know. And there's so much mysteries. And it's actually a lot of fun just to know that there's so much out there that needs to be explored. That's really inspiring, both for sort of our urge to do more research, but also inspiring for the next generation, because there's so many questions that you can come up with, and the answers can always be improved. We can always know more, and so that's exciting. Anyone else?
I can add to that, just sort of anecdotal, how have I changed? Or I guess I've been a neuroscientist for a while, so it's kind of hard to say. But, when I watch my kids learn how to walk, for example, it really brought back all the memories of learning about, how the spinal cord neurons are firing and they're developing central motor pattern generators. So it's really interesting once you start thinking about neuroscience and how your brain it's an organ that really integrates outside information into your body. I think it really changes it at the fundamental level, how you interact with the world and how you see the world and how you see everyone else, right? This is how we understand our external environment. So it definitely changed me tremendously.
I would chime in also to say that I have found that learning about the amygdala has been extremely important in my relationships with my colleagues, with my family, and it's really fun to begin to use your neuroscience knowledge as power and how you can dominate the world. I mean, it's really fun if you understand things about like feedback loops that you might get into and so forth, that you can actually use it to help you in just your everyday life.
One thing my colleague warned me when I first is, don't treat this as a neuroscience experiment. It's really easy to use the power of reinforcement learning, all these amygdala pathway when the kids are growing up. But one thing that my colleague said is just enjoy being parents and switch off the neuroscientist in me, and that's actually fun to know. But the next question, actually, it's at least inspired from my kids getting stuck in the home. Shikan asked the question, does the effect from addiction differ in the case of substance addiction versus addiction to things like sugar, or even abstract things like gaming, binging movies? If yes, how? My kids are stuck with Animal Crossing right now at home.
So yeah, I'm going to take that one. And I think KC kind of highlights actually one of the points that I was going to make. And that is, I think we need to make a distinction between true sort of compulsive inability to control behavior, sort of what we think of as an addiction, and sort of that more casual use of that term, addicted. So I think a lot of us, I'm so addicted to sugar, to chocolate cake, or I'm addicted to gaming, that sort of thing, where you're just really into something. And in that sense, I would argue that that type of sort of us being really interested in something and loving it is separate from addiction. Addiction's really a pathological, compulsive behavior that we can't control, right? And so in that sense, there are subsets of individuals that do have true addiction to things like sugar, as well as gaming. I'm not so sure about the binge movie watching, but exercise. I mean, all kinds of shopping. There's a shopping addiction. So again, those aren't just like, oh, every time I get a paycheck, I run out and I, go buy a new outfit. That's different than people that will spend, hours and hours and hours on the internet making purchases to the point of going, hundreds of $1,000s in debt. And, in that sense, those folks, the gaming addictions, there's some evidence that, some sorts of obesity and overeating becomes sort of a compulsion type of thing. And so in that sense, their brains are going to be very similar in the way that people with substance disorders have an addiction to drugs, and that in that there's those profound, long-lasting changes in the brain that drive decision-making, motivation, that sort of thing. What we don't know why, so don't ask this question, is we don't know why some of those similar brain areas and how they change, why that makes one person sort of develop an addiction to a drug, and while another person might develop an addiction to exercise, another to sugar, and that sort of thing. So I think it's pretty, interesting area of research, but we don't know that. The other thing I wanted to mention that people might be interested in, sort of speaking to this, there is a really interesting finding and happening that occurs in people with Parkinson's disease. So a lot of addiction is regulated by a neurotransmitter in the brain called dopamine. And it turns out that you've got these folks that have Parkinson's disease, and Parkinson's disease is marked by a depletion of dopamine. And often when we actually see those, profound symptoms, they have greater than 90% loss of dopamine cells in their brain. And so one of the main treatments for people with Parkinson's disease is to give them a drug called L-Dopa, which is basically a dopamine replacement therapy. And in a small subset of these people, they develop a pathological gambling condition. And it's really interesting because these people have never gambled in their life, and many of them are in their 70s and their 80s, and all of a sudden they, likely because of this treatment, they start having a gambling addiction where they'll gamble away everything. And so, it's again pretty interesting. I think it gives, maybe not interesting for them and their families, that's another issue, but from a science perspective, it's really interesting to try to understand, well, what is it about that dopamine and what's happening in their brains that all of a sudden give them this addiction. Okay, I'll turn it over now, back to Casey.
And hi, we'll just do a brief interpreter switch again. And we're good to go.
Okay, so this is a shout out to Jackie and Holly, who's doing amazing work interpreting our ASL language interpreter, and it's something to shout out for all the organizers, making this accessible to communities that use ASL as well. So the next question, I thought I'd bring that up, submitted by Jacqueline. The sign language, is it processed in the same part of the brain as auditory language? So I think I'm closest to this area. I would think that language is actually processed in an area, it's actually under the temporal lobe underneath. It's not necessarily auditory, it's multisensory. So the fact that when you read, when you have phonemes, so the sound conversion there, and in fact sign language also, because it's a language, it has units, and I believe they are called visemes. So we have visual visemes for our mouth, but there are also units of passing in sign language, and I believe that's also processed in not auditory language per se, but language in general. That's sort of my take on that question. Okay, so the next one is, Sarah, are you back? Oh, we've lost Sarah again, so I'm going to jump
I'm
to
back.
You're back. Awesome. Sarah, so Terry asked a question, curious to hear your take on the ethics of neuroenhancements.
Yeah, I think it's a great question, and it's the sort of topic that always comes up in philosophical discussions about neural interventions, or any interventions for that matter. And, I think going back to something we touched on earlier, a big question here is it really an enhancement? So there are things that might appear to be enhancing that aren't really. And I think the example of many of us, especially as we begin to age, would like to have a bit stronger of a memory, but it's not always true that having more is better of something, right? And so we could have more and more and more memory. And as Zen said before, that there's a value to forgetting some of the things that we experience. And so we want to make sure that when we're thinking about what might be changing our bodies in ways that aren't really just treating something that's a problem, but really enhancing beyond what's typical, that it really is truly an enhancement. And then beyond that, I think there's also this important question about, if in fact we really figure out something that's enhancing, how do we deal with the kind of justice and distributive issues around making sure that access is available? Because if it truly is an enhancement, the world that we live in, most of us, right, is one where we can get a pretty radical divide between people who can afford access to those things and people who cannot. And we can just exacerbate some of the justice problems that we have if we're not very carefully paying attention to those things. I think, even as we think about where to focus our priorities, or as we think about materials that go into them, or, how we develop the devices or pharmaceuticals or other things that could in fact be enhancing. But that doesn't really do it justice. I also think there's a question, once people get access to something that's enhancing, how do you ensure that people really have a choice to say, no, it's not for me? Because we end up in a workplace scenario right now where we need certain kinds of devices and access in order to even keep a job. And, we might think that in the best possible world, we're able to choose and actually say we prefer not to do that. But in at least some social circumstances, it becomes nearly impossible to make that unpopular choice.
All right, thank you, Sarah. So I thought that this would be a fun question to bring everyone in, submitted by Chloe. What is each of the panelists' favorite, fun, surprising fact about the brain? So my fun fact is, a lot of times when we say primary sensory cortexes, the old thinking is the auditory cortex only process auditory signals and visual the same. That's actually no longer the case. That's how we view it. And thank goodness that is not the case, because we study multisensory processing and the cross-wiring of sensory information in many different parts of the brain, including what it's coined as primary sensory cortexes, is actually really important for our everyday lives. So that's my fun, surprising fact.
My fun, surprising fact is that it's possible to change people's activity rhythms purely with the color of light, not with any intensity. You can just do it strictly by color. And that's why the color of the sky is different at sunrise and sunset. And this is one of our evolutionary ways of detecting the time of day.
Any other takers on this fun, surprising fact about the brain?
I feel like I'm not the person to take this question, so the neuroscientist can correct me if I'm wrong about what my fun, surprising fact is. But just from relatively recent work on memory, this idea that every time we go back to a memory, we're in fact altering that in some way. I think that's a really interesting idea, because the sort of old view of how we store memories and just kind of, tape drive, although obviously that's not the medium, is just inaccurate, right? There's a way in which it's an ongoing process. And so we're switching those things every time we're consulting them.
Oh, awesome. So let's move on to the next question. Jaden has a question. How does the increasing role of technology in learning change our brain structure or how we learn? Susan, you want to take on this?
Yeah, so I was going to answer this in a couple of different ways. So actually, one of the areas that we study in the lab is how technology and what we think of as sort of sensory stimulation, how that affects young kids. And so we know that there's been an increase in diseases like ADHD. There's been an increase in diagnoses over time that doesn't really track on to how to genetic factors, right? So it has more to do with sort of environmental influences. And so I've paired up with Dr. Dimitri Christakis at Children's, and as well as Nino Ramirez. And Dimitri is a pediatrician who studies sort of development of ADHD-like syndrome in kids as a result of being exposed to all sorts of media devices as they are developing. So I would answer this question in a couple of different ways. So during early development, when a child's brain is still developing, we think that the sensory stimulation, so the lights and the tones and the visual stimulation that you're getting, is really sort of fundamentally reorganizing sensory input into the brain. And so that doesn't so much speak to how learning might change, but one of the interesting things that Dimitri found is that because the sensory stimulation is sort of reshaping the brain early on, what might sort of appear as an ADHD-like disease later in life is sort of a result of kids now going into a classroom and having to sit very still and listen to a teacher and not have all of that sensory stimulation that they're used to when they're on their iPad and getting lots of different devices. And so so that's one sort of interesting take on that question. The other thing it's been a really interesting study I remember reading about that suggests that maybe seeing something on a device isn't really quite the same as seeing it in person. So they did this study where they had kids watch someone on a device open a window and put like a stuffed animal or something through the window, and then the kids had to go find the stuffed animal. And they watched it on a device, and then they watched it in real life. And the kids that watched it in real life were able to sort of replicate that behavior and find the stuffed animal, whereas the kids that watched the video of it weren't as good at performing the task and retrieving the stuffed animal. So it kind of seems that in-person interaction is maybe more valuable than seeing that same interaction, or is maybe a better learning experience than seeing it on a device. But I guess all of that being said, I think it's also important that, the reality is technology and devices are fundamental parts of our world and are not going away. And so, I think it's very interesting from a research perspective to kind of understand how they might be changing, but I think they also open up a lot of different opportunities. So, this being an example, perhaps there can be an audience from all across the country that can now listen to these questions and ask us questions. Whereas if we were just doing this in person, it would be just whoever could come to the auditorium that day. And so I think technology opens up a lot of access to materials that you wouldn't otherwise have in your life. And so it can facilitate learning in that sense.
I want to add to Susan's experiment, quoting that whether you learn it from the screen versus in live actors, where you can find the stuffed animals. And a lot of parents cite these type of studies as a way of saying, let's not have any screen time at all for the kids. I think that's actually really taking that experiment's interpretation a bit too far. I think it's about really spending the time with your kids with the technology. So the technology can definitely, it's not a replacement, but it can be an enhancement when it's used when you have joint attention with the kids. So I'm often sort
Absolutely.
of taken aside when parents said, "Oh, I don't want to participate in your experiments because of screen time." That's actually not the deficit. It's really when you can do this together and have that quality time with the kids, and then it changes the nature of how technology is used.
Can I chime in here? The
Please.
one thing I would caution is that there's a lot of evidence that increased screen time is one of the major contributors to the development of pathological myopia, because we now get high spatial frequency information on our retina that we never evolved to have. And it is a signal for the eye to grow.
Wow, I'm going to tell that to my 7-year-old daughter, who is having problems with with short-sightedness.
Yeah, that's interesting.
That is really interesting. I didn't know that.
Yeah, thank you, Maureen. The next question submitted by Caitlin. As a neuroscience researcher, are you allowed to change subfields in neuroscience? Could one switch from psychopharmacology to schizophrenia research? I, for one, started as an engineer, so I not just even subfield, a whole discipline. I'll let others chime in.
I would chime in and say, you're allowed to switch from anything to anything in life. It's your life. You got to do what your heart tells you to do. I started as a molecular biologist. My PhD is in biochemistry and molecular biology, and I switched to neuroscience, as in vision, as a postdoc. But I both of those parts of my life are with me today. So you can always switch, and it's really fun to mix these things up, and that's how a lot of progress is made, is by people in one area suddenly getting interested in another area and then bringing the two areas together.
Cross-pollination is definitely needed, and it's a plus for any fields, any subfields. We only gain from it. Okay, I think we have an interpreter switch right now.
Yes, interpreter switch. And we're good to go. Thank you.
All right, Maureen, might have you back for this one. Roland Dover asked, do blind people undergo REM sleep?
To my knowledge, they do. They undergo everything. The thing is, the blindness is a relative term because you're legally defined as blind when you're 2200. At that point you're legally blind, but but people that are legally blind still see. They have light perception. They can see a lot of things when they're very close up. That's why they can migrate around the world with a white cane or a dog. They can see a fair amount. It's just that they have to get very close to objects to see more detail about it. They typically have circadian rhythms, and REM sleep, as far as I know, is not affected. Circadian rhythms sometimes can be, but you have to be very advanced in terms of retinal degeneration to lose your ability to have circadian rhythms.
Funny that I should talk about switching of attention. I'm trying to monitor the different questions and switching from one screen to another. It's cognitive taxing. The next question's from Madeline. How does environmental factors in a young child's life affect how they learn and pick up speech traits, and can they be changed or discouraged later in life? So I don't know whether you know Professor Higgins in the My Fair Lady musical. I always want to meet such a person to decipher my accents. So I grew up in Hong Kong, moved to Sydney, went to Boston. So I packed my car before I went to Harvard Yard, and now in Seattle. So there are language as you pick up, especially early in life. And we know that even perception of different languages. So the co-directors of Institute for Learning and Brain Sciences, where I'm based, Dr. Patricia Kuhl, looks into sort of when there's a sort of a learning period there's a window of opportunity for you to really get language and understand these phonemes. So when it's before 6 months, you're sort of citizens of the world. You can sort of get all the different phonemes, and then after the critical period, after 9 or 10 months, you're sort of locked into the statistics of the language that you get most exposed to. And so actually raising your kids with bilingualism or multilingualism, it's a good way to ensure that you still have the statistical model of the different languages. Once you miss that window, it's much harder to pick up different traits as you get older. It's not that there's no neuroplasticity. There will always be. I will pick up a different accent, I'm sure, if you get me to South Africa, for example, for a few years. But it's more from it has a lasting impression when you're younger in age. Okay.
I would just chime in here and say that the visual system has a critical period as well for certain features of it. And if you don't have visual input, it seems totally different than language, but it's not. It's the same thing. If you don't have visual input in a certain window of time, for example, stereo vision, if you have a misalignment of the eyes, strabismus or amblyopia, if you don't get that corrected early enough, you never develop normal depth perception. If you have a congenital cataract and have it corrected as an adult, your retina sees just fine, your brain sees nothing. So critical periods of development are a really interesting thing in the sensory system.
What about the Prakash project by blanking my name from their vision scientists at MIT? They go and remove cataract for young kids, and they can still do visual scene analysis though, right?
So if you do it with kids, it's fine. If you do it in an adult, if you do it in an adult, if someone has a congenital cataract and never get visual input, even though their retina works perfectly normally, they can't see.
Awesome. All right, so the next question from CIELT17. What question do you most want to find the answer to during your time as a neuroscientist? Does anyone want to have a go at that?
How the retinal circuitry is one of the most beautiful things on earth. I mean, it is just gorgeous. And we now have these technologies, serial block phase scanning electron microscopy, that allows us to take an entire volume of retina and recreate the neurons, their anatomy, their connectivity, and try to understand how this gives us vision and what aspects of vision. We have motion, we have edge detection, we have color. Just taking something that seems so simple as the retina and trying to understand the circuitry and just the art of the retina.
All right. Susan, I believe you want to have a go at the next question. Have there been times when an experiment or study was scrapped in the design stage due to concern of violating ethical guidelines? Oh,
no, I thought
Susan just texted me saying that it's a cool question. Let me.
It was the next one. Sorry, the next one about synesthesias. I was thinking Sarah might want to answer that question about the ethical concerns around deciding when experiments could be conducted or not.
Or have there been experiments that needs to be scrapped in the design stage? Oh, I think we've
Did we end up losing her?
Did we lose Sarah? Okay, we might come back to that.
Okay. Well, actually, so this is Susan and my dog. I guess one thing that I thought of around this question, I didn't so much know about something being scrapped, but the other aspect of things, I was thinking about these studies from Helen Mayberg, who does, she looks at treatment-resistant depression, and she had done some work with a company around implanting deep brain simulators to help these folks with with treatment-resistant depression. And the study was conducted. It was funded. I'm not sure if it was funded privately or with the government, but so the study was completed. These devices actually were shown to be very efficacious. So they were very good at treating this this depression. And as I'm sure everybody knows that has any sort of technological device, over time, those devices stop working, right? They don't, maybe the battery runs out, they need to update, all of those sorts of things. And there was a lot of ethics regarding what happens at the termination of these studies when there's no longer funding for that. You've now got a bunch of patients with these devices, and they're basically like, who's going to pay to have these supported? So I thought that was kind of an interesting, so on the back end of sort of an ethical dilemma about once we've done something to a person from a research perspective, and then is it unethical not to in some way maintain the kinds of support that we gave them basically during the experiment? Do we have do the ethics go beyond when the experiment ends? And so I thought that was kind of interesting. I think in those particular cases, the researcher, Helen Mayberg, she actually made sure that she was able to secure additional funding for those particular patients in order to support their devices, which was also kind of cool. And I'll turn it back to you, Casey. I guess so Casey, are you on or muted? Oh, I think you're muted.
I'm sorry, I was muted. I was just commenting on the surprises you get from live streaming, including not unmuting yourself. Next question from Valerie: how does synesthesia work? Is it the result of too many connections between neurons, and if so, how does that happen? Maureen, do you know anything about synesthesia?
I have listened to a number of lectures on synesthesia, and I do not understand how it works. I am sorry to report.
It's a fascinating question indeed. Would love to know the answer. So the next one from Julia: why do headaches make people extra sensitive to sensory input, especially light and sound? I recently had that experience myself. That was really annoying. I had to switch off all the lights. Maureen, do you know anything from the light sensation part? Maybe I can have a go at the sound part.
No, I mean, I don't understand why people have it. People definitely have photophobia. There are a lot of different kinds of retinal diseases associated with phobia, but I personally don't really understand a lot about the causes of photophobia. I mean, I understand the retinal things that are happening, but I don't know why they're having so that the light seems so bright that they can't take it, but I don't understand it beyond that.
Yeah, there are many different ways in the auditory world too. So you can have hyperacusis, where you're just hypersensitive to sound, but you can also be in a situation where it's too overwhelming, when there are too many sounds, but you can't make sense out of it. And so that could also be sensitive to just being overwhelmed. But whether that associated with headaches in general or some other sort of cognitive process that's being overwhelmed, I'm not sure. Let me finish up with the last question by Jacqueline. And I believe she says, don't quote me on that, but Susan said something along the line: how does addiction affect the structure of the brain? Was it the frontal lobe?
Wow, exciting that I get to finish this off. So yeah, so the frontal lobe, the frontal cortex, that is our decision-making area of the brain. And so, again, this depends a little bit on the different kinds of drugs that we're talking about, but the pyramidal cells that actually Zen mentioned at the beginning of this, those get rewired, and they change their structure as a result of drug use. And what many researchers believe is that if you think about the frontal cortex sort of being able to, that's our inhibitory circuit, right? So that is the circuit that tells us, this isn't a great idea to do something. And what happens with drugs is we lose that inability to be able to say that something's a bad idea. And what's likely the case for that is that we get changes in dopamine D2 receptor. So that dopamine I was talking to you about before that can regulate motivation, that can act on these dopamine receptors, and those dopamine D2 receptors, we get a decrease in their activity over time. And those are also located in that frontal cortex. And so as a result of that, there's sort of two things that happen. When we are exposed, when a person with a substance use disorder sees a cue that's associated with a drug, then that they get a strong activity in areas like the cortex. And then the part of the cortex that would tell them, well, it's not a good idea to go seek out this drug, that activity is suppressed, and then that's what can drive the drug use. So I'll end there. I don't know if KC had just general wrap-up thoughts, but I'll turn it back over to him.
Well, it's 9:00. I believe this is the first time I've ever been involved in a live stream, and I have been utterly amazed by the organization that the Evening with Neuroscientists team has put on, especially Lila working in the background. And so I want to bring Lila back, and so that we can all thank her and the invitations for all of us and the panelists to join you tonight. I have a lot of fun, and I'm pretty sure that the rest of the panelists have been enjoying this opportunity to talk to you as well. So Lila says, oh, Lila, would you like to join back?
Yes, hi. I would just like to say that it's been a real honor working with you, and I know I connected with all of you sometime around maybe November last year. It's been a long time. And so, and there's been a lot of ups and downs with, oh no, this isn't happening. It's still happening. Maybe it's happening. And every time we've done this, the technology has not cooperated with us. So it's been really sort of a sense of pride now that our team has just pushed through all of the technological issues and bring this to everyone. And so that's pretty much what I want to highlight, just my team, all the people working behind the scenes, designing all of the art, doing all of the art, and people who've put in 6 months of work to then quickly convert it in just 1 month to make this happen.
Well done. I think we all enjoyed it, and hopefully everyone enjoyed it on YouTube. So I think we'll be signing off. Thank you for joining us tonight. Good night.
Thank you.
Everyone.
It's been fun.
Thanks.
Yep. Thank you.